Hi class, and welcome to chapter 2, which will be on the parts of a cell. And this will help you label some of your cell pictures in your lab packet that you need to fill out for this module. These are several important scientists that made really important discoveries about cells. Cells are the smallest living units in the body. They obtain nutrients, make molecules needed to survive. They dispose of all our wastes. They maintain their shape, and they're able to replicate themselves and divide. There's organelles within cells that are subunits of cells with specific functions. So just like we have organs within our body, cells have these tiny organs or organelles. Most cells contain the same basic organelles. Not all cells have the same in abundance, but for the most part, a lot of them have the same. Cells have three main components. The plasma membrane is the outer boundary. The cytoplasm contains most organelles, and the nucleus is like the brain, controlling all cellular activities. So this is a great slide to pause, use it to fill out your lab packet, and we'll be going over all of the organelles, all the structures within the cell in this lecture. The plasma membrane defines the extent of the cell, so it keeps things inside the cell, which we call intracellular fluid, from the extracellular fluid outside and between cells. The structure of the membrane is what we call a fluid mosaic model made up of a lipid bilayer, so two layers of lipids. There's different types of membrane proteins. The integral proteins are firmly embedded or attached to the lipid bilayer. You have short chains of carbohydrates that are attached to these integral proteins. And then the peripheral proteins are attached to the membrane surface, which support the plasma membrane from the cytoplasmic side. And this is a look at the plasma membrane according to the fluid mosaic model. You can see the integral proteins expand throughout the entire surface of the membrane, whereas the peripheral membrane proteins more just touch the outer surface, either exterior or internally. The functions of the plasma membrane are to relate to the location at the interface of the cell's exterior and interior. The plasma membrane provides a barrier against substances outside the cell. Some plasma membranes will act as a receptor, and the plasma membrane basically determines which substances can enter or leave the cell because the membrane is selectively permeable, so it only allows certain things to enter and certain things to exit. To get to these animation videos, you need to download the PowerPoint off of Canvas, and then you're able to play the animation. So I'm going to use these animation videos within these PowerPoints as a way to cover some of the topics. So please download and play this animation video on membrane structure. Membrane transport then either moves through simple diffusion or osmosis. Simple diffusion is the tendency of molecules to move down their concentration gradient from an area of high concentration to low concentration. And osmosis is the simple movement or diffusion of water molecules across a membrane. And here's a look at simple diffusion, things moving from an area of high to low concentration. Osmosis is the diffusion of water, and water gets through your plasma membrane by going through what we call an aquaporin. Facilitated diffusion uses a carrier protein to help things get through from an area of high to low concentration. So all of these processes on 1, 2, 3, they don't require any energy, but active transport is moving something from an area of low concentration to high concentration, and that always will require energy, also known as ATP. Facilitated diffusion is the movement of molecules down their concentration gradient through an integral protein, and then active transport uses integral proteins to move molecules across the plasma membrane against their concentration gradient. Endocytosis is a mechanism by which particles can enter your cells through phagocytosis, cell eating larger solid particles, or penocytosis, which means cell drinking, anything that's a fluid or liquid. Receptor-mediated endocytosis is where the plasma membrane, plasma proteins will bind to certain molecules. It will make kind of a coating, a pit that forms the vesicle, and this will be taken into the cell. And this is how insulin, some hormones, enzymes, and low-density lipoproteins enter your cells. So here's a look at phagocytosis, things entering the cell that are large and solid. Penocytosis is smaller fluid-containing solutes. And then receptor-mediated endocytosis are when the solutes bind to receptors and then the receptors form a vesicle around the thing that they want to get into the cell. Exocytosis then is a mechanism that moves substances out of the cell. The substance is enclosed in a vesicle. The vesicle migrates to the plasma membrane. As shown here, the vesicle kind of fuses with the plasma membrane, and the vesicle contents are released to the exterior. And here's a photomicrograph of a secretory vesicle releasing its contents at about 100,000 times magnified. So then we'll get into the cells. The cytoplasm lies internal to the plasma membrane, and this consists of the cytosol, the organelles, and all the inclusions. The cytosol is the jelly-like fluid in which all cellular components are suspended, and this can consist of water, ions, and enzymes. Ribosomes are constructed of proteins and ribosomal RNA. Ribosomes create proteins. So if there's one thing you remember about ribosomes, ribosomes create proteins. The assembly of proteins is called translation, and the ribosomes are like the assembly line of the manufacturing plant that creates proteins. Ribosomes can be free-floating, and they will be within the cytosol, or ribosomes can be attached to the endoplasmic reticulum, which is what we'll talk about. And these make proteins to renew the plasma membrane and make proteins that are exported from the cell. The endoplasmic reticulum is the system of membrane-walled envelopes and tubes throughout the cytoplasm. And we can have smooth or rough ER, is what we abbreviate it as. Rough ER has ribosomes, and smooth ER consists of tubules and a branching network. It doesn't have any ribosomes attached to smooth ER, so no protein synthesis is made in smooth ER, only in rough ER. So here's a look at what ribosomes look like, whether they are rough ER, so studded with ribosomes, and then smooth ER will not have the ribosomes attached, shown here. The Golgi apparatus is a stack of 3 to 10 disc-shaped cisterns, which sorts products of rough ER and sends them to the proper destination. Products of the rough ER move through the Golgi from the convex side to the concave side, and the Golgi apparatus is like your Amazon distribution center. It packages and then ships out where it needs to go, either inside the cell or outside the cell. And this is a look at the Golgi apparatus, the vesicle entering on the cis face, the receiving side, and then it's being repackaged, reformulated, and then exited out on the trans face, which is the shipping side. So here's a look at the sequence of events from protein synthesis on the rough ER to the final distribution of these proteins. And you can pause the slide and read through these steps on your own. Lysosomes are membrane-walled sacs containing digestive enzymes to help digest unwanted substances. They're the cell's demolition crew, you could say. And this is what a lysosome looks like. The light green area are the regions where the materials are being digested. A mitochondria is the power plant of the cell. So mitochondria, power plant, the big thing I want you to know about mitochondria is that it creates ATP. This is the source of energy for the cell. Your mitochondria will release energy stored in chemical bonds, transfer the energy to produce ATP, and the mitochondria are surrounded by double-walled membranes. Cells with high energy requirements will have more mitochondria, like your muscle cells, your cells in your brain. Your mitochondria are the most complex organelle. They contain some maternally inherited DNA, and they are believed to have arisen from bacteria. So this is a look at mitochondria with a double membraned wall, the outer mitochondrial membrane, inner mitochondrial membrane, and then the cristae and matrix. You'll learn how the mitochondria function when you get to physiology. The peroxisomes are membrane-walled sacs of oxidase or catalase enzymes. These basically are the toxic waste removal system of your cell. They neutralize free radicals, break down poisons. So we'll see them in your liver and kidneys. The cytoskeleton then, this question is your case study question for this module. It asks you to describe and list the 3 types of cytoskeleton or cell skeleton network. And these are the 3 types. So if you're listening, go ahead and pause this slide and start filling out your case study. Microfilaments, intermediate filaments, and microtubules. And I'm going to let you guys research these 3 on your own for your case study and have you learn the material that way. And you can see pictures of each, so you should be able to describe what they do and what they form. and which one is the largest and which one's the smallest. The centrosome and centrioles, so your centrosome is the spherical structure in the cytoplasm, and centrioles are the paired cylindrical bundles that act in forming cilia, flagella, and the mitotic spindle, which help in cell division. Cytoplasmic inclusions are temporary structures. They may consist of lipid droplets, crystals of protein, or glycosomes. The nucleus then is the control center of the cell. It's like the brain. It controls all cellular activities with DNA. DNA is what provides instructions for protein synthesis, which is extremely important. And the nucleus is approximately 5 micrometers in diameter. So here's a look at the nucleus, the brain. It has pores within the nucleus where things can enter and exit. The nuclear envelope are 2 parallel membranes separated by the fluid-filled space. Nuclear pores penetrate the nuclear envelope, which allow large molecules to pass in and out of the nucleus. The nucleolus is the little nucleus in the center of the nucleus that contains parts of several chromosomes, and this is where the ribosomes will be kind of created and put together. DNA is made up of a double helix that's made of 4 subunits. They can be thymine, adenine, cytosine, and guanine, and DNA will be packed with protein molecules that form chromatin. Each cluster of DNA and the histone proteins that keep it together is called a nucleosome. And this is a structure of DNA, a double helix wound molecule made up of these bases that pair together. So guanine always pairs with cytosine, thymine pairs with adenine, and so forth. The extended chromatin is the active region of DNA where DNA's genetic code is copied into mRNA called transcription. So transcription is just the copying of the DNA genetic code into mRNA, and condensed chromatin are tightly coiled nucleosomes, which are inactive. Chromosomes are the highest level of organization. They contain a long molecule of DNA, and a typical human has 46 chromosomes. So here's a look at how DNA is tightly wound together around histones to form a nucleosome. The nucleosomes form condensed chromatin. The chromatin forms very, very condensed chromosomes. And again, the chromosome is the kind of typical X structure that you think of. And the chromosomes are in the extremely tightly wound DNA. The cell cycle is the series of changes that a cell undergoes. Interphase is the growth phase, where the cell is metabolically active, it makes proteins, it grows, and the centrioles will start to replicate near the end of this G1 or growth phase. The S synthetic phase is when DNA will replicate itself to get ready to kind of pull itself apart for cell division. And the G2 phase are where the centrioles finish copying themselves. During all 3 phases, the cells will carry out its normal metabolic activities, and this is interphase. Interphase is where your cell spends about 90% of the time. Now we'll talk about mitotic phase, which is cellular division, where the cell spends about 10% of the time. The mitotic phase follows interphase, and this is where your cell divides. Cell division involves mitosis, which is the division of the nucleus, and cytokinesis, which is the separation of one cell into 2. So mitosis is divided into 4 phases. Prophase is the first phase. This is where the chromatin will condense into chromosomes. In late prophase, the centrioles will move away from each other and the nuclear membrane will dissolve. So here we have interphase, normal cell growth, early and late prophase. We have early prophase. The DNA is replicating. Late prophase, we don't see the cellular or the nuclear envelope anymore around the nucleus. Metaphase is the second phase of mitosis. The big thing is that the chromosomes will be in the middle of the cell along the equator. Anaphase is the third and shortest phase, and this is where the chromosomes will split. So we have metaphase and anaphase. Metaphase is easily seen because the chromosomes are lined up in the middle of the cell. And then anaphase, you can see all the chromosomes being pulled apart from each other by the centrioles and the mitotic spindle. And then telophase and cytokinesis, we start to see a cleavage furrow, which will form eventually where a new plasma membrane will form to separate one cell into 2. So telophase, we had the chromosomal movement stopping, the nuclear envelope will form again, and then cytokinesis is just the division of the cell into two daughter cells. Great animation video on mitosis. Please watch these as they help to supplement the learning. Development aspects of cells, specialized functions of cells relate to their shape and their arrangement of organelles. Cells that connect body parts or cover organs are fibroblasts, epithelial cells, erythrocytes, are red blood cells, which take up oxygen. So here's a look at fibroblasts, epithelial cells, and erythrocytes. They work to connect body parts, connect body parts. Epithelial cells form linings, and erythrocytes transport oxygen and carbon dioxide. Cells that move organs and body parts are skeletal and smooth muscle cells. So here's a look at a skeletal muscle cell and a smooth muscle cell that help to move organs and body parts. Cells that store nutrients are fat cells. Cells that fight diseases are macrophages. Here's a fat cell. It's filled up with adipose, which is fat, a lipid droplet. And here's a macrophage to help fight off diseases. Cells that gather information are neurons, which are long processes for receiving and transmitting messages. So here's a look at a nerve cell. It gathers information and controls body functions. We'll go over more of this in depth when we get to the nervous system. Cells of reproduction, sperms are in male. They have a long tail for swimming to try to get to the egg for fertilization. Shown here, a sperm cell. The sperm cell is the only human cell that has a flagella. Aging is a complex process caused by a variety of factors due to free radical theory, which is damage from byproducts of cellular metabolism, where radicals will build up and eventually damage cells. The mitochondrial theory is just that the decrease in the production of energy by mitochondria weakens and ages our cells. The genetic theory proposes that aging is programmed by genes, where telomeres are structures that limit the maximum number of times a cell can divide, and telomerase prevents telomers from degrading. And that's chapter 2. Thanks for listening, guys, and we'll see you for chapter 3. Please note that you do have a quiz on chapters 1 and 2 in module 1.